It's an absolute pleasure to introduce Dr. Venugopal. Not only is he a good friend, he's a trusted source of information. He's been with Optum, which is a group previously known as Healthcare Partners. Now we've joined the Optum group, which gives us a much larger footprint nationwide. But he's been with Optum as a hospitalist for at least the last 6 years. He's very well qualified to give us the talk he's about to give us. He's doubly board certified. He's board certified in internal medicine, as well as fellowship trained in infectious diseases. And because of his expertise, he's the chair of the Optum Vaccine Advisory Committee, and he's the director of infectious disease for our group. So certainly very well spoken, very well qualified. In Holy Cross, he's also no slacker. He's on the Antibiotic Stewardship Committee. He's on the Infectious Disease Committee, Sepsis Committee, and most recently he was elected to the MEC as a member at large. I've heard him talk many times. He's updated us on where we are in vaccines, where we are with the virus, and I'm always learning something. So I appreciate the talk, and I'm looking forward to it. Anil, are you on?
Yeah, I am. Hopefully you guys can see me. Thanks, Dr. Farid, for the introduction. So today I'm going to try to give you an update and just make some sense of the SARS-CoV-2 virus, the vaccines we have available, and just talk a little bit more about the variants that they've been talking about in the media, so it all makes a little bit more sense. So just to start off with the basics of microbiology, viruses by themselves, they actually can't replicate. So they are either DNA or RNA entities, and they have either envelopes or they're non-enveloped viruses. But the main thing about them is they actually require the host to replicate. So essentially, once they infect the host, that's how they make more copies of themselves. So if you follow this little diagram at the bottom here, once the virus enters into the host cell, you can see that the DNA or the RNA gets entered into the cell, and then it uses the host machinery to make copies of itself. And then those copies get envelopes again, and then they can go on and infect more and more cells. And that's how the virus kind of propagates. So what are coronaviruses? So coronaviruses are basically a large family of viruses that predominantly cause disease in animals. So sometimes they can have mutations, and when those mutations occur, they can jump and cause infections in humans. And there's really 7 different types of viruses, coronaviruses, that can actually cause infections in humans, and they're predominantly cause respiratory illnesses. The 2 big ones that we probably knew about before SARS-CoV-2 was the SARS virus and then the MERS virus, which is the Middle East Respiratory Syndrome. Both of those had caused epidemics in the last 10 years. And because of those, we actually had a lot of information about this virus before we even had this pandemic. So talking a little bit about the basics of the virus. So there's a lot of different components to the virus, and it's made up of different proteins and enzymes. Essentially, it's a single-stranded RNA virus, and it has these little red spike proteins on it. And these spike proteins are really important because these spike proteins are what bind to the receptors on the host cell, so then it's able to enter into the cell and then cause infection. So just a little bit about mutations. You know, anytime any cell is really dividing and making copies, there's always a chance for mutations. And when you look at the different types of cells or bacteria or microbes, you can see that depending on the size of the genome, we can see that the rates of mutations are higher. So the smaller the genome, the more chance there is of having copying errors and leading to mutations. Whereas in like human cells or in bacteria, which have much larger genomes, you see the mutation rate is much smaller. So again, anytime there's a copying error when the copies are being made, that's what's called a mutation, right? When you have a group of coronaviruses that share the same inherited set of distinctive mutations, that's when we talk about it as a variant. And then if there's enough clear-cut differences in this group, it can actually be called a strain subsequently. And again, this is just a diagram of that same picture showing the different components of the virus with the RNA in there. And then this, again, is the spike protein that would bind to the ACE2 receptor, which is present on the host cell, so it could enter the cell and make copies. So when we talk a little bit about mutations, normally, you start off with this one parent virus. And again, every time the virus replicates, there's always a chance for mutations. Most of the time, these mutations are actually deleterious or defective, and they really don't actually help the mutant. But every now and then, the mutant actually gets a selective advantage, and that advantage could be anything from being more resistant to certain therapeutics, or having the ability to spread more quickly, or being able to evade immunity. So when that selective pressure is put on there, so say this mutant here is resistant to certain medications, if you suddenly introduce that medication, obviously it's going to wipe out all these other copies, and then this mutant is going to start to propagate. So when we talk about SARS-CoV-2 variants, there's been a lot of talk about this, just because what happened was initially in the pandemic, we had this wild-type virus that was spreading, and then as they slowly kept an eye on it, they started to notice that over time, we've been having increasing numbers of variants that have developed. The WHO has really defined these variants based on how serious they are, basically. So it's considered a variant of interest if there's a cluster of cases that seem to be driving a surge. So if they're doing sequencing and they notice that a certain variant has developed, but they're not quite sure about it, then they call it one of interest. Once it's known to definitely cause more severe disease or be more contagious or potentially be resistant to medications, that's when they call it a variant of concern. And then when it gets to the point that it actually causes more severe disease and hospitalizations, that's when it gets to the point of calling it a variant of high consequence. And fortunately, so far, there are no variants of high consequence with the SARS-CoV-2 virus or the COVID-19 virus. But you can see that just in a really short time frame, from the beginning of January to the end of February, which is really just 2 months, the breakdown or the predominance of the different strains, you've seen a real big increase in the variants that are of most concern. And there's really 5 variants of concern. There's the B1429, the 427, the 17, the B1351, and then the P1 variant here. So looking at the US, they really only type and look at the different types of variants in about less than 3% of the actual number of positive samples that are collected throughout the US. But you can see that the predominant one that we see is the B117, and then it's followed by the B1351 and then the P1. And this is generally speaking across the US. And you can see that even the reported number of cases, considering we've had millions and millions of cases, it's still a very, very small number. When you look at the same distribution of these 3 viruses globally, it's very similar. The most predominant one seems to be this B117, which was originally identified in the UK. Then there's the B1351, which is the one that was originally identified in South Africa. And then the P1 variant, which is believed to have originated in Brazil. So what are the implications? Well, when you have a variant, again, those mutations can cause a selective advantage. So it might cause it either being able to spread more quickly. It might be able to cause either more mild or severe disease. It might be able to evade detection by specific diagnostic tests. It could have decreased susceptibility to certain agents, including monoclonal antibodies. Or what's most concerning is if that mutation actually allows it to evade natural or vaccine-induced immunity. So normally when you're exposed to a virus, part of the immune response is the development of antibodies, and that's what's represented here on the left side by these little diagrams. Once you're re-exposed to the virus, the antibodies in the person that's been exposed should come out, and then they come and neutralize the virus. But in the case of a variant, if there's been a mutation in one of these proteins on the surface, it might not allow for full neutralization of the virus. So even though you've been previously infected and have technically an immune response, because this is a variant, you might still actually get sick from the virus. Now, talking about vaccines, because I know a lot of people want to talk about this. Generally speaking, the whole vaccine process is a very long, drawn-out period. Usually it takes anywhere from 4 years to 30 years to develop a vaccine, and you can see that just based on there's multiple stages that usually are required to go through in order to get to the final product here. A lot of times, again, it can take years just to get to the point where you're doing animal testing, and then it can take 5 to 7 years in human trials, and then you submit the information to the FDA, and then assuming they approve it, you can get the vaccine manufactured. This can take, this takes a lot of money and a lot of time generally, and often similar to drug development, about 7 out of 8 vaccines actually fail. They never actually make it to approval because they don't actually do what they're supposed to do. question becomes like, how were we able to make the SARS-CoV-2 vaccine in such a short time frame? Well, a lot of that is just by circumstance. We had a lot of existing information on the preclinical from the SARS and the MERS viruses that we knew from before. They were able to use some of that pre-existing data from animal studies and then do animal studies very quickly. And then on top of that, there was a big cash infusion from the US government. And what that did was it allowed them to start running these multiple phase trials in parallel, which is because they had less risk, because the pharmaceutical companies didn't have to worry about running out of money or if it failed, because they already had the backing of the US government. And at the same time, they were able to start producing in mass quantities before they even submitted to the FDA. So that's how we were able to get to a vaccine in 10 months. So as far as vaccine development goes, as of March 4th, at least a few weeks ago, you can see there's dozens and dozens of vaccines that are being developed across the world globally, and they're all in different phases of development. These phases 1 through 3 being the human trials. But right now, at least in the US, there's only 3 vaccines that have been authorized. I don't want to go into too much detail on this, but there's really multiple platforms that can be used to how the vaccine actually works to produce the immunity. Some of these platforms have been used before in other vaccines, like these viral vector replicating vaccines, the live attenuated, which we've seen in like flu vaccines, inactivated vaccines, which we've used in flu vaccines, protein subunits been used in vaccines like Shingrix, which is for herpes zoster. But the ones that are currently authorized, this viral vector vaccine that's non-replicating and the mRNA vaccines, these actually previously didn't have a vaccine that was already authorized for use in humans for other diseases. So that's why there was a lot of uncertainty about this. But one of the things about these techniques is that they were able to produce a vaccine much faster than you can with a lot of these older techniques. So there's really 5 vaccines that are really far along as far as phase 3 or late stage trials in the US. 3 of these have already been authorized by the FDA, which includes Moderna, Pfizer, and Janssen's vaccine. AstraZeneca is already finished their phase 3 trial, and they've actually been authorized in other countries, including by the WHO. And then Novavax, which is a US company, has finished their phase 3 trial, and they already finished a phase 3 trial in the UK, and they should be submitting for their emergency use authorization in the next month or 2. So it gets really challenging when we talk about these vaccines, because a lot of times people want to draw comparisons and everybody thinks, okay, well, you can just say, well, this is this percent and this is this percent, so this one must be better than that one. But that's not always the case. And the reason is, like, because this was done in the span of 1 year, these trials were actually run in different populations with different protocols, and there was actually different circulating variants. So to try to compare the vaccine efficacy of these is very difficult because they're not the same conditions. These vaccines weren't basically studied against each other. To put things in comparison, when we talk about vaccine efficacy, basically when we talk about flu vaccines, we're really excited when the flu vaccine efficacy is anywhere from 40 to 60%. To see this kind of vaccine efficacy is actually really impressive. But what's more important than this is not all vaccines will actually prevent disease, right? A lot of them basically allow you to have an attenuated disease, so you don't get as sick as you normally would. And that's what is the key to these vaccines right now. When you look at the protection of these vaccines against severe COVID-19, so that's when people get so sick that they actually require being on oxygen, or they get hospitalized because of these vaccines, you can see that all of these vaccines are very, very protective against this. And that's why you don't want to just get hung up on these numbers. You have to look at kind of the big picture. So when you talk about which is the best vaccine, realistically, the best vaccine is the one that you can get. And just to summarize, really there's 3 currently FDA authorized vaccines, and there's probably 2 more that are really close on the horizon, with multiple more in different or like later stage trials. The current vaccines, so mRNA vaccines and the non-replicating viral vector vaccines like the Janssen vaccine, they're not live vaccines, so they cannot give you the infection. So we know that they're safe to be used in more susceptible populations. All 3 vaccines are very effective at preventing severe COVID-19 or hospitalizations. There's currently 5 main variants out there, and there's a risk that they might be easier to spread or even cause reinfections in people that were previously infected or vaccinated. Again, the best vaccine is the one that you can get. And then right now, based on what we know, regardless of whether you've been vaccinated or not, you really have to continue to follow all the mitigation strategies. So try to minimize improve your social distancing, continue to follow good hand hygiene, make sure you're masking, at least until we have enough information about how likely you are to spread infection once you have been vaccinated. And that is it. So any questions?
Jen, do you want to ask your question that's in the chat?
Sure. I was just wondering if there's a way to predict at any point, is there any predictive modeling on how many variants can be in play at a particular time, or is it a total just chaotic situation? You can't predict it.
I really think the chaotic situation, because theoretically the mutations occur every time there's an infection and the virus is replicated. So it's just a matter of, does that variant or that mutated virus actually become the predominant virus and start spreading to others? But yeah, I don't think there's any minimum number of variants that can be out there, really.
No algorithm can save us then?
I don't think so, no.
Okay, thanks.
I don't actually have the chat box open, so if there's questions in there, I can't see them.
I think Arif has a question.
Yeah, I got a question, thank you. Is there any difference between the length of time this particular virus can live outside the body on inanimate surfaces compared to other viruses? Are there any changes?
I haven't looked at this data in a while, honestly. I don't think it doesn't have a prolonged period outside the body, that's for sure. And I know that because, they have done studies looking at how it can survive in the environment, but I don't think it's any significantly different than other viruses like, say, flu or any other respiratory viruses.
Anil, I was wondering if you could project as to what sort of side effects people may see down the line beyond the immediate ones in regards to these vaccines. I think that's the biggest mystery. Has there been any discussion around that?
So, it's a very valid question, and that's probably the disadvantage we have with producing vaccines so quickly, is that when you looked at those other timelines that I showed you, when we do things over years, we actually have really long-term follow-up on these patients, usually several years of data to say, okay, they got the vaccine and then the immune response persisted and they didn't suddenly grow a third arm or anything like that. We don't really have a lot of information about that. As far as again, the mRNA vaccines really didn't have a platform that was used in humans, at least in as a commercial vaccine. It had been clinically tried for other diseases, but didn't actually work. So I don't think we just know enough, honestly. The adenovirus, the viral vector vaccines, we have used those as replicating viral vector vaccines, which is that same platform that Janssen's using has actually been used for the production of the Ebola vaccine. So we have a lot of data on that, in more than 190,000 people that have been vaccinated. But I don't think they've seen any concerning trends, otherwise they really wouldn't have went forward with it.
So for those that are weary of the mRNA formulation, they should pursue maybe that formula?
That's an option. The important thing with the mRNA vaccine is to understand, so it doesn't actually interact at all with the host DNA, right? The mRNA actually gets reproduced in the cytoplasm, whereas the DNA of the host is actually in the nucleus of cells. So there's no interaction. And then on top of that, it actually has a pretty short half-life. So that mRNA gets destroyed within a couple days after they've been vaccinated. But that short time frame is enough for them to produce the proteins to stimulate the immune system or the antigens essentially to develop the antibody response against. So it's not like that mRNA is persisting in you for weeks or months or years or anything like that. It gets broken down pretty quickly just by the normal host cells within a very short time frame. So, I don't anticipate there's going to be anything long-term down the line, but who knows? Honestly, I can't really predict, unfortunately.
Right. Bobby, did you have a question?
Yeah, just a quick question. In regards to the side effects or long-term, just following up on Marla's question, as far as I understand currently, and I think traditionally, the reporting of side effects, adverse events, or deaths is a voluntary method. Is that correct?
Yeah, theoretically, yeah. Because I mean, essentially, somebody has to identify that they got the vaccine and report it. There's multiple systems right now in place for this. So, I mean, they encourage people to sign up for this thing called VSafe. But then there's the VAERS system, which, again,
The VAERS system is a voluntary system, right?
it is correct. And, like, there's no mandatory way because there's not really a way to track this many millions of people, right? And that's why they did what they can during the clinical trials. But you're right, after that, they'll follow those people in the clinical trials for a couple of years. But the people that have gotten it since then already, unless they report it, you're right, we're not going to know about it. But that's really how it works with all vaccines. Most things are picked up in clinical trials, but every now and then there might be a signal that occurs where somebody starts reporting things that happen later on, and then they say, oh, there seems to be a trend towards this, and people that are getting vaccinated with it. But again, you're right, it is voluntary.
Right.
Does anybody want to share their thoughts on this presentation, or like, not whether it's good or bad, but really like what your reactions are to what you heard? Is that new information? Is it different information? Are you hearing different? As Dr. Klein always says, are you hearing other rumors on the street?
Yeah.
I thought it was excellent, and I thank you, Anil. It was really good for me.
That's Dr. Perrine, so he's a tough critic, so that's good.
No, I thought it was
we
great.
all get a copy of this, by the way.
Absolutely.
Yeah, it was an amazing presentation. What is Optum doing in regards to their vaccination program? I know Kaiser's really put a program together. Providence has. What's Optum doing? I haven't heard any news around that.
So we're pretty spread out in Southern California, right? We're actually spread out, I think it's 7 different public health regions. So there's like 5 counties and 2 cities that we're in. So we had actually set up around 18 clinics, vaccination clinics in those multiple sites. And I think we've vaccinated a little bit over I think it's close to 50,000 patients at this point.
Wow.
That's despite having really limited access to vaccines like everybody else. So we're doing what we can, and then we're starting to work with the new supplier now with Blue Shield, and then we'll start vaccinating outside of Optum patients and into the community as well.
Which vaccines are you offering from Optum?
So I think predominantly we've been getting Moderna, but some sites have Moderna. And I don't know if we've gotten Janssen yet, honestly, because I don't know if there's been a large shipment into California.
Yeah, I was wondering, do we know, Bernie, I don't know if you know, are we going to be getting the Johnson & Johnson by chance?
Yeah, it's my understanding we will at some point. We just don't know when.
Okay.
I mean, there's a great advantage with that Janssen vaccine is the refrigeration. The cold chain is a lot easier to maintain. So we're hoping to get that Janssen vaccine because since it's a single dose and you can get pretty good vaccine efficacy, it'd be a lot easier to roll that out for like our homebound patients. So we're planning on instituting a program for that once we're able to, for the people that can't actually make it to these sites to get vaccinated but are actually high risk.
Right. I that would be fabulous.
Yeah.
I just had a question. Somebody texted. Do we have any idea how often, or if we'll have to get revaccinated? Do we think? Is that the big question?
Yeah.
And like, part of the thing is like we don't know because like these clinical trials, a lot of the Moderna and the Pfizer one, they literally finished their because. What was required by the FDA was for them to have a median 2-month follow-up of these people after they got the second dose of the vaccine. So those clinical trials were only running for like 4 or 5 months, really. But by the time these drugs got authorized, we haven't even really got the full data from them on how robust is the immune response, and therefore we don't have long-term follow-up on like how long do these antibodies actually stick around. Said that, like the full immune response isn't just based on antibodies, there's also like a T cell response, so which isn't measured in some of the commercial tests that are currently available. So we just don't have enough information to say, like, is it going to be an annual thing or not? And then, like, what's going to happen? Are these vaccines going to hold up against the variants? There's just, like everything COVID related, there's just a lot of unknowns still.
Well,
Anil, in regards to just a quick question about the compared to the regular flu vaccine, do we know how long that stays in the system? I know we get the flu vaccine currently annually, right?
Correct. Yeah. Generally, because the flu season essentially runs from November through April, and you have a pretty good immune response with the flu vaccine for at least 6 months, which is why usually you only get vaccinated once during the flu season. But because there's always like a different strain circulating, and then they tweak the vaccine formula to account for those different strains that were being predicted for the upcoming season. That's why we have to do it. Not quite sure whether that's going to happen with COVID or not.
So, Anil, I have a question. I'm sure they're tracking this, but would there have been, or is there any value to getting and I know you say there's different parameters to look at your immune response. Is there any value to getting, let's say, an antibody level prior to getting the vaccine, then getting it, let's say if you're getting Pfizer or Moderna, 10 or 14 days after that second dose, just to see what your antibody level is, and then getting it, I don't know, 3 months, 6 months down the road to see if it's staying where it's supposed to be, or what level it would need to go down to, in theory, I suppose, to see if you need a booster. Is there any? They're accumulating data on this, I suspect, but they've been around, a few months now. Are they getting any preliminary results?
So, I mean, there's value in doing that in the setting of a clinical trial. And the reason I'm saying that is because, even collecting data, like say we vaccinated 100 people here in Holy Cross, and then we were like, hey, we're going to follow them. Like, the problem is a lot of the tests now commercially available are qualitative tests, so they just say it's positive or negative for the antibody. And keep in mind, those different antibodies are available. Some will actually tell you whether you've been infected. Some will tell you whether it could be the vaccine or it could be infection. But we don't know, like, what's that threshold.
Okay, all right. You've answered the question. Yeah, that's good.
So they are collecting data on those people, and I'm hoping they'll give us information. Eventually, they'll be able to say, this is the number that needs to be there to be protective. But we just don't have that yet, honestly.
Okay, all right. That answers it. Thank you.
I was going to also ask you, just from your perspective, I mean, for us in the hospital, it really seems like we've had such a downward rapid decline. Are there any projections that we may have to deal with another surge?
I mean, it's definitely a possibility. If you follow some of these modeling, like the Institute of Health Metrics and Evaluation, I think it's out of University of Washington, and even some of the other ones on the CDC site, some of them suggested we were going to have a surge again in towards the spring, but as we've been moving forward, those numbers have slowly been dropping, those predictions. I think that's largely an effect because of the vaccines appear to be very effective. And then so far, people have been maintaining all those other mitigation strategies. What we're seeing in other states, it doesn't appear to be because of variants that we're starting to see spikes in other states. It appears to be because they've started easing off of a lot of the restrictions that were put in place, and people are starting to get out in public and socialize again without proper precautions, and that's why we're seeing more transmission.